Thank you everyone for being here tonight. We'll go ahead and get started. So good evening, everyone, and welcome to the latest installment of the Torrey Botanical Society's Torrey Talks lecture series. My name is Catherine Mercier. I'm the Society Programming Chair. To start, I'd like to share a little bit about the Society. So the Torrey Botanical Society was founded in New York City in 1867 and is an organization for anybody interested in plant life, including professional and amateur botanists, students, and anyone who simply enjoys nature. The objectives of the Society today are to promote an interest in botany and to collect and disseminate information on all phases of plant science. To that end, we hold meetings, field trips, public lectures like these. We have our journal. We fund graduate student research and education, and we also sponsor symposia and regional conferences. The best way to stay updated on future lectures, the field trips, and everything else that the Society is doing is to become a member, which you can find out more about on our website, www.torreybotanical.org. You can also follow us on social media. All of the social media handles are up on this slide. We are at Torrey Botanical almost everywhere on X, Facebook, Instagram, and Blue Sky, and on YouTube, where we post the recordings of these lectures. We are at Torrey Botanical Society. Before we begin the talk, I want to highlight some upcoming events. So next week, next Friday, we have our annual banquet at the New York Botanical Garden. This starts at 5 o'clock. The talk will be at 5:30. And the talk is going to be given by Dr. Emily Sessa, who's the director of the Herbarium at the New York Botanical Garden. And her talk will be titled Specimen Tales, Stories and Insights from the William and Linda Steer Herbarium. After the talk, there will be a dinner in the rotunda at the library. So we'll be at the library at the New York Botanical Garden. All the information is on the website, torreybotanical.org, where you can find tickets. And then that weekend, we have our first field trip of the spring. Is it spring? At Alley Pond Park, skunk cabbages and bryophytes of Alley Pond Park. And that's at 10 a.m. At Alley Pond Park. And again, to register, go to the website, torreybotanical.org. And finally, so we are in the middle of our lecture series. This is our March talk. Next month, we have a talk from Ashley, Creativity at the Forefront of Understanding Art, Science, and Ourselves as Entangled Necessities as a World in Transition. So keep an eye out for an email announcement for registration for that talk. And just a couple notes about Zoom before we begin the talk. Please keep yourself muted during the talk, but you're welcome to have your camera on or off. When we reach the Q&A portion of the talk, you can either raise your hand and unmute yourself to ask your question, or put your question in the chat to be read out. And after the talk, if you would like to share the talk or rewatch it, it will be recorded and available on the YouTube page. So I'm going to stop sharing and introduce our speaker for tonight, Alfredo Justo, who is currently the Curator of Botany and Mycology at the New Brunswick Museum, where he oversees the curation and growth of the NBM Herbarium. This herbarium has over 100,000 specimens of plants, fungi, lichens, bryophytes, and algae. Alfredo received his PhD from the University of Vigo in Spain, working in part on the diversity of Pluteaceae in the Iberian Peninsula, which we're going to hear more about tonight. His research merges morphology-based and molecular methods to study the origin, evolution, and current status of fungal diversity, both locally, the fungal diversity of Atlantic Canada nearby, and he also has ongoing global taxonomic research on selected genera of mushrooms. And with that introduction, I will hand it over to Alfredo to talk about the family Pluteaceae. Basidiomycota and Agaricales in North America, a decade of molecular studies.
Thank you, Katie, for the introduction. Can you hear me okay? Everything
Yes,
okay?
we can hear
Everything
you.
looking good? Okay. Thank you, Katie, and the organizers for inviting me to give this talk. I'm going to tell you, we're going to veer a little bit off of the botanical into the mycological, and we're going to talk about one particular family of mushrooms, of mushroom-forming fungi, the family Pluteaceae, which is one of the groups that I studied for my PhD a long, long time ago. So I've been hanging out with these guys for over 25 years now. And I'm going to try to focus on things related to North America and what we know about the family in North America. First of all, as Katie mentioned, I'm the curator of the New Brunswick Museum, which is the provincial museum in New Brunswick, Canada. We're located in St. John, right by the beautiful Bay of Fundy. The NBM Herbarium, which is just a small part of what the museum is, has over 100,000 specimens of plants, lichens, fungi. The fungal part is really the one that we are trying to build up. And that's one of the reasons when the previous curator retired, they were looking specifically for a mycologist to really build that part of the collection. And we focus on New Brunswick as the provincial museum, but we also keep an eye in general for collections from Atlantic Canada and Eastern North America in general. We have a very small but very active team of people. This is Amanda Brebner and Kendra Driscoll, who work with me at the herbarium, and we try to integrate and modernize the herbarium as much as possible, while at the same time taking care of public programming and research at the museum. And this is just a small part of what I actually do for research. Even though I'm the curator of the whole collection and I take care of all the parts of the collection, my research really focuses on mushroom diversity. What species of mushrooms do we have in New Brunswick? By extension, what do we have in Atlantic Canada and Eastern North America? And I want to mention that most of the work that you're going to be seeing today is not my work. There is no way of doing this work alone. So it's not my work alone. This is the result of many years of collaboration with both professional mycologists and citizen scientists, both in Europe and North America. And you're going to see when I present some of the publications, you're going to see some of these names again listed as the co-authors in the publications. But really, without big collaborations with many people in different parts of the world, this type of research that looks at diversity at broader scales wouldn't be possible. So even though I'm presented today, without them, this work wouldn't have been possible. This is just a little summary of what the talk is going to look like. I'm going to give you first a little bit of an introduction of what the family Pluteaceae is and the results from the first phylogenetic studies that were done more than 10 years ago. Then most of the talk is going to focus on the taxonomic work that we have been doing on the family Pluteaceae in the last 10 years. And again, I'm going to focus the result mostly in the North American species. And I'm going to finish the talk by giving you a small preview of ongoing projects that are happening right now. So the Pluteaceae is a relatively small family. It's in the Agaricales. The Agaricales is one of the largest orders of mushroom-forming fungi. It includes most of the more typical mushrooms that you're going to see out there in the field, the ones that have a cap, they have gills underneath, they have a stem. That's the most typical morphology in the Agaricales, even though there are many other morphologies. The family Pluteaceae itself, these are saprotrophs. These are decayers of organic matter, in most cases wood, but in some cases other substrates. And they have a combination of characteristics that are rather unique among mushrooms. They have pink spores. It's not something that you're going to see when you put the spores under the microscope on the individual spores, but you can see the pink color on the gills here is because the spores are maturing. If you do a spore print of this mushroom, the color is going to be pink, which is already a pretty unusual character among mushroom forming fungi. There are lots of mushrooms with white spores or dark spores, brown spores. There are not that many with pink spores. The gills are free. The gills that you see here never reach the stem. That's also a characteristic that is not very common. And then they have a very unique characteristic to the family, which is the inverse lamella trama. What is the lamella trama? If you take one of these gills here, gill or lamella, and you make a section and you put it under the microscope, how are the cells of that trama arranged? In many cases, they're going to be parallel to each other or slightly irregular. In the case of the Pluteaceae, they have a very particular way of arrangement, which is the one that is highlighted here, that is called inverse lamella trama. It's called inverse because the ends or the hyphae are pointing towards the inside of the trama, which is a very, very unusual characteristic. And the family, because of this combination of characteristics, in traditional morphological taxonomy, was always a neat, well-defined little group that was very easy to characterize because nothing else would have this combination of characteristics. And it had only two genera, the genus that gives the name to the family, Pluteaceae, that you can see right here, usually saprotrophic on wood directly, and Bulbariella. And the main difference, they both have these characteristics, and the main difference between the genera would be that Bulbariella, as the name says, has this membrane at the base of the stem that is called a universal veil or a bulba in mycological terminology. And Pluteaceae would not have that membrane at the base of the stem, but all other characteristics were really just the same for both genera. There was another genus in traditional taxonomy that was sometimes mentioned. It was the genus Chamaeota. And Chamaeota was basically a Pluteaceae that had a partial veil, or what we call in mycology, a ring. This veil, instead of being at the base of the stem, will be somewhere in the stem, covering the gills when they're young, and then it will remain on the stem as the mushroom grows. But even from morphology alone, most people could tell that this genus, Chamaeota, was pretty much just a Pluteaceae with a ring. So some people will just say, oh, maybe just this character is not enough to really consider it a separate genus. So before I did my first really big phylogeny of this genus, which was my first postdoctoral project, my PhD back in Spain was morphology only, was traditional taxonomy, looking at this group from using traditional morphological techniques. And when I did my postdoc in the States at Clark University in Massachusetts, I was looking at this group from the molecular perspective. Before I did that, there were a couple of studies that included some samples in their phylogenies to look at the relationships of Bulbariella and Pluteaceae. And one of the first big studies of the Agaricales show Bulbariella and Pluteaceae as distantly related, which was, again, was very odd because the family Pluteaceae was so characteristic in terms of morphology, and the genera really have very few differences to each other, that it was odd that in the phylogenies, these groups ended up in very distant places. And then there was another study in 2006 where Bulbariella and Pluteaceae were actually recovered as sister taxa. So we had very contradicting results in the early phylogenies of this group. So in the year 2010, I was focused on generating data, getting everybody that I knew that was working on Pluteaceae or Bulbariella. We had people from the US, we had people from Russia, we had people from Brazil, from Mexico, from Italy. So I was trying to get anybody that I knew was actively working on this genera. And in 2011, we published the first big phylogeny just focused on the Pluteaceae. And I should mention any of the publications that I mentioned today, they're available on my webpage. So you can just go there and download the PDFs if you want to read more about this later. Obviously, today I can only give you the highlights for many of these things. So in this first phylogeny, remember, we had these preliminary results that were saying basically two opposite things, that Pluteaceae and Bulbariella were related to each other, and they were saying that they were distant to each other. What we did find in the phylogeny is that they both were correct, and that the problem was that Bulbariella, as we have conceived the genus in terms of morphology, was actually two separate lineages, very different from each other, very far away from each other in the phylogeny. Pluteaceae itself, that you see highlighted here, is monophyletic. That had no problem. Now, Bulbariella, the core of the genus, most of the species were actually not related to Pluteaceae at all. But there was a small group of species of Bulbariella that were actually the sister taxon to Pluteaceae. And these species that ended up in the phylogeny next to Pluteaceae, they had mostly characteristics of Bulbariella. They had that bulba at the base of the stem, that universal veil, but they were actually in the phylogeny next to Pluteaceae. So that group ended up with a new name, with a new genus, and it was a combination of the both genera that existed in the family up to that point. It was named Bulbopluteus, which basically means it's a Pluteus with universal veil, with a bulba. So what are the characteristics of Bulbopluteus? The genus is different from Bulbariella in the structure of the cells of the cap. How are they organized? In Bulbariella, the cells of the cap are really broad, and in Pluteaceae, the cells of the cap are really thin. The hyphae on the cap are really thin, and they're also gelatinized. They're embedded in a matrix that under the microscope looks like jello, and that's typical of Bulbopluteus. That is not present in Bulbariella. And another characteristic is the length of the spores. In Bulbopluteus, the average spore length is always above 11 microns, while in Bulbariella, it's always below 11 microns. This genus that we created, Bulbopluteus, was also different from Pluteus first, because it has that universal veil at the base of the stem, and also almost every single Pluteus species has spores that are also smaller, that have less than 11 microns in length. The structure of the cells of the cap, what is called the pillae pellis in mycological terminology, in most species of Pluteus is actually also different from Bulbopluteus, but there are a couple of species that can actually have a similar type of pillae pellis than in Bulbopluteus. So the separation there was not as neat as with Bulbopluteus versus Bulbariella. So we solved that mystery. We solved the mystery of these apparently contradicting early results of Bulbariella ending up in two different places, because there is the true Bulbariellas that are not related to Pluteus, and there is a small group of things that used to be in Bulbariella that now they're gonna get their own genus, Bulbopluteus. And in this case, and this is not something that happens often in mushroom-forming fungi, the morphology was also helping. The morphology was also lining up with what we were seeing in the phylogeny, which many of you are botanists or imagine what mycologists, we have had to do taxonomy just looking at the fruitings of the organism. Imagine doing all the botany classification just looking at fruits, never looking at the plants. That's what mycologists before the molecular era had to do, figuring out the relationships just looking at the fruiting structures. Because even if we could look at the organism, at the mycelia itself doesn't have a lot of morphological characters. So most of the taxonomy of mushroom-forming fungi up until the molecular era was based on the morphological characteristics of the fruiting structures, which are the mushrooms. Bulbopluteus itself is a pretty small genus. When we look at the phylogeny in these early phylogenies in 2011, we recognize 4 species. These two, Bulbopluteus gliocephalus and Bulbopluteus erle, they were previously classified in Bulbariella, so we just transfer them to Bulbopluteus. This species, Bulbopluteus asiaticus, was actually described as new from Japan. The original collection was identified as Bulbopluteus gliocephalus, but you can see molecularly is quite different. And another good thing about this is that the species actually, you can also separate them using morphology. It gets a little bit trickier. There is sometimes some overlapping characters. It's not perfect, but we were able to construct a dichotomous key that people could use to identify the species, which in the molecular era for mushrooms is not always possible. Sometimes all you have is the actual differences in the DNA sequences. So there is an actual morphological dichotomous key that you can use. And I'm going to stop for a second on this species highlighted here. As you can guess from the name, Bulbopluteus michiganensis, it was originally described from Michigan in the US. But it was described in 1934, and the collection that we have here in the phylogeny, that's the type. The 32 makes reference to the year. This was collected in 1932. It was described by one of the more important mycologists of the 20th century in North America, Alexander Smith. And he actually described this species as Pluteaceae michiganensis, which is weird because it does have that universal veil at the end of the stem. And Alexander Smith himself noticed that something was weird about this species of Pluteaceae because the spores were really large. And he makes that comment in the original publication from 1934. The largest spores are unusual for the genus Pluteaceae. And what's really curious about this species is that Alexander Smith himself doesn't mention the species ever again. And he did a lot of work on Pluteaceae, but he doesn't mention his own species that he created ever again for the rest of his career. There is no mention of Pluteaceae michiganensis. What we have guessed over time is that probably he realized, oh, this is actually not a Pluteaceae, it's probably a Bulbariella, and he forgot about the species altogether and never thought about it again. But we were actually able to sequence the type, and that Pluteaceae michiganensis that he described almost 100 years ago is actually a Bulbopluteus, as the spore size would indicate. And if you look at the type collection, it actually has that universal veil at the base of the stem. And we were able to place it in a phylogeny, which is not bad for molecular techniques. We did this in 2010, and the type specimen is from 1932. And we did get a barcode for the type collection, which is really, really great. So we were able to resurrect an old name, a forgotten name that has been absent from the literature of North America for a species originally described from North America, from Michigan. Pluteaceae itself is going to be the focus of my talk because it's the more diverse genus. Now that we have figured out the problem with Bulbariella, there is actually two different genera, one Bulbariella, one Bulbopluteus. Pluteaceae itself is where most of the diversity is. Previous estimates have estimated the diversity of the genus between 150, 300 species. I can tell you right now from the phylogenetic data sets that we have already estimated 250 species. And there are whole parts of the world that are simply unsampled at this point. So probably the total worldwide diversity of the genus is probably going to be around 400, 500 species. And Pluteaceae, the genus has three sections, three groups that are morphologically defined depending on how the cells on the cap are organized and what type of cystidia of sterile cells they have on the gills where the spores are being produced. So if the cells on the cap are just elongated hyphae parallel to each other, and on the gills, they have cystidia with thick walls and hooks at the very apex, you're looking at section Pluteaceae. If the cells on the cap are rounded and the cystidia have thin walls and no hooks at the end, then you're looking at section Celluloderma. If the cells on the cap are elongated but upright, perpendicular to the surface, and the cystidia have thin walls, then you're looking at section Hispidoderma. And this classification with the small variations has been maintained before the molecular era. And surprisingly enough, it holds up pretty well when you look at the phylogeny. There are actually, when you do phylogenies in Pluteus, you get the same 3 lineages. And you can look at the evolution of the cells in the cap, and you can look at the evolution of the cystidia. And for the most part, it makes sense. It lines up with the morphology. There are a couple of groups that kind of are the exception to the rule. There's always going to be some exception. But I would say for 90% of the species of Pluteus, looking at morphology, you can guess where that is going to end up in the phylogeny in terms of section, and you're going to be right. What we also did and what we started doing is looking at the species level diversity. How many species do we have? Are the estimates of species diversity of Pluteus based on morphology correct? So in our first phylogenies, this is again 2011, we had 98 ITS sequences. ITS is the internal transcribed spacer, is the barcode locus for fungi. It wasn't officially the barcode by them, but it's now the official barcode. And we had pretty limited sampling. The collections that we had in our first phylogenies were mostly from Western Europe, from Eastern and Central US, a little bit from Japan, a little bit from South America, but they were really skewed in terms of geographical representation. But we could already tell, just looking at the phylogenies, we could recognize about 25 species or species complexes of Pluteus present in North America. The problems were, well, first of all, ITS alone, even though it's actually a pretty decent barcode, but it has limitations. It's not going to give you all the answers that you want all the time. And another problem is it was very difficult sometimes to establish correspondence between the clades, the lineages that we were seeing in the phylogenies. This is one of our early phylogenies, and the older names that were available for Pluteus species. And you're going to see a couple of examples why that is difficult in the talk today. So that's it for the introductory part. Now I'm going to get you through the travels through the diversity of Pluteus that we have been doing for the last 10 years. And then I'm going to finish up with some ongoing projects. So the first thing that we did is to focus on the more commonly recorded species of Pluteus, Pluteus cervinus. If you're going to see one species of Pluteus in a field guide for mushrooms, it's going to be this one, Pluteus cervinus, called the deer mushroom. It was originally described from Europe, but it has been reported all over the world. And in Europe, there were other species that were described as similar or closely related to Pluteus cervinus, depending on the taxonomic concepts, between 12 and 15 species. And in North America, the same thing. Pluteus cervinus is reported, plus additional species. And sometimes the same species are reported in Europe and North America. So we wanted to know, well, if we focus just on this group, and this is Pluteus section Pluteus, Pluteus cervinus, the more commonly recorded species, if we just focus on this, what picture of diversity we're going to get? And again, this was a collaboration. We had people from Eastern North America, Western North America, Europe, a lot of collections that cover the whole geographic span of Russia. We have over 300 collections, and we were trying to put all the pieces of the puzzle together. We were looking at the morphology, both the external morphology and all the characteristics that you look under the microscope, the spores, the cystidia. We're looking at the ecology. In Pluteus, sometimes it's important if these things are growing on conifer wood versus angiosperms. We included a lot of collections from North America, and we knew that the barcode, the ITS, was not going to be enough. So we selected a second locus, in this case, a single copy protein coding gene that was shown before to be a use for fungal taxonomy, TF1. And we tried to put all those pieces of the puzzle together. And the puzzle became much more clearer. This is the paper that we published in Phytotaxa in 2014. And we were looking at the taxonomy of this species group across the Arctic region. And we were able to identify 26 species in total occurring in Europe, Asia, North America. 12 of those species were described as new in 2014. So up until 10 years ago, a lot of the species that now we know they exist, we were not even aware that they were here. And they're the ones that are highlighted with the red dot in the phylogeny here. In North America, if you make the comparison, it's even more dramatic. We have, or we presented 19 species in that paper. 10 of them were described as new. So half of the diversity of the North American species was not known, was not named until up 10 years ago. And one of the problems, and this is not unique of Pluteus, it's very common with mushroom-forming fungi. Again, we're trying to do taxonomy just looking at fruiting structures. It's going to be misleading. We have a limited number of characters. This is something that when I do live this presentation of a version of this presentation live, I try to trick people into telling me how many species do they think are represented in these pictures. The picture on the left shows a lot of morphological diversity. You have white mushrooms, you have orangey mushrooms, gray, with a lot of fibers. And the picture on the right shows very little morphological diversity. So if we were to do a taxonomy without any other information, we would end up with the wrong conclusions, because the picture on the left is actually just one species. This is all the variation that you can get in one species of this group. This is actually Pluteus cervinus. And every single photo, every single collection on your right represents one individual species, so 6 species in total. So morphology alone, if you don't have the complement of the phylogenetic data, is gonna, most of the time, is gonna mislead you. One thing that we were able to establish, and that has been shown for many other groups of mushrooms, is that the idea with mushrooms and with microorganisms is that everything is everywhere. These are just widespread species, and the reality is very different. We have endemic species at the regional level. And in North America, for the most part, the Eastern North American endemics and the Western North American endemics are different from each other. These are all 5 species that we described. They're endemic to Eastern North America. And these are 3 species that we describe, or that were described before us, that are endemic to Western North America. So that concept that endemism was something that other groups had, but not fungi, it really has fallen apart in the molecular area. We do have either regional or continental endemics in fungi. One thing that was also important, and I always like to stress, is like, without participation and collaboration with citizen scientists, with people from mycological societies, people that do mycology as a hobby because they like mushrooms, they like to study these things. Without them, some of the species would have never been found, or it would take even longer to find them. This is Pluteus orestes, which is a species from the Western North American mountain ranges, like Sierra Nevada and similar mountain ranges there. All the collections from these species that we use to actually describe the species were made by citizen scientists doing different projects in mostly Yosemite National Park in California, but also some other mountain areas out west. Without those citizen scientists, we wouldn't have found this species, at least not for our 2014 paper. And these are all the observations. If you go to iNaturalist and you search for Pluteus orestes, this is the known range of the species on planet Earth. This is what we know about Pluteus orestes. It's a species of Pluteus that occurs associated with conifers at high elevation in the mountain regions of Western North America. This observation here that marks the southernmost confirmed occurrence of the species is a very recent iNaturalist observation. And many of these species have sequence data that citizen scientists are generating in collaboration with professional scientists, or sometimes on their own. And even for section Pluteus, that we did all this work in 2014, the work is not done. We're still gonna be finding more species, even in North America, that we sample quite well for our study. This is some Pluteus collections that Terry Clemens that you see here. Terry is a non-professional mycologist, and she lives in Arizona. And she collaborates with the people at the University of Arizona there, but she's not a mycologist by training. And she's doing like regional inventories of fungi, and she has found already a new species of Pluteus. This is going to be described hopefully in the next couple of months. This is Pluteus arizonicus, and it's a species that occurs, again, associated with conifers, high elevation forest, and it's probably more widespread in the Southwest, but currently it's only known from Arizona. So that was just 1/3 of the story. That was section Pluteus. But as I told you before, there are two whole other sections, lineages in Pluteus, and what happens with those. We repeated the process. We repeated the formula. Collaboration, collaboration, collaboration. Get citizen scientists involved. We cannot be everywhere collecting everything. And they're also interested to know what their collections are. Our phylogenies for these groups, for the other two sections, are now over 2,000 collections of Pluteus. And these are projects that have been building up for the last 10, 15 years. And again, we're trying to put all the pieces of the taxonomic puzzle together. Morphology, ecology, geography. And again, we know that the barcode is only going to take us so far. So we're doing 2 data sets for the phylogenies. And this is where the diversity is. Like section Celluloderma, you saw that Pluteus have 26 species. Section Celluloderma has 155 species. And again, this is mostly focused temperate boreal areas, northern hemisphere. There are whole parts of the planet that are not sampled for this group. Section Celluloderma also quite diverse, up to 61 species in the phylogeny so far. And the last piece of the taxonomic puzzle is what happens with all the species of Pluteus that have been described before we have PCR machines, before we have DNA, before we have phylogenies. In North America, there have been quite a few species of Pluteus described by 19th century and early 20th century mycologists, especially Muriel. He described 46 species of Pluteus from North America. Peck also described 10 species, and Atkinson described 6 species. And most of the type collections exist somewhere in herbaria. For Muriel, just focusing on what Muriel did, there's out of the 46 species that he described, 17 were already evaluated before we started doing phylogenies, and 8 names that Muriel described about more than 100 years ago. 8 of those names kind of survive the molecular process, and we know what those names mean. We are able to connect that name with a certain clade, with a certain lineage in the phylogenies that we're building. But there are also quite a few other species that have not been evaluated yet. And this is something that we did as part of this ongoing process. Muriel described species mostly from the Northeast. He was based in New York for quite some time, and then he moved to Florida. Then he described quite a few species from the southern states, and he did a couple of trips to the West Coast, so there are also some Western species from Muriel. So as part of this process, we asked for all the type collections that are hosted at the New York Botanical Garden and at the University of Florida Herbarium. And these collections, the type collections and other original material, they have been revised already in the literature. Smith, Alexander Smith, that I mentioned before with Pluteus michiganensis, Singer, other mycologists of the 20th century have revised these collections. The problem is, even though they were supposedly looking at the same collection, they didn't always make the same descriptions, and they didn't always came to the same taxonomic conclusions. And an additional problem is that for many of these species, these type collections and the few additional original material that might exist are the only collections there are for that species. There are very few species in North America herbaria that use the names described by Muriel, by Peck, by Atkinson. So it's very difficult to get a sense because not only we have very few original collections, but also very few collections that use a modern concept of that species. And we're, at the end of the day, what we're trying to do is to connect those names to the clades that we see in the phylogeny, see if those names can be revived, can be resurrected and used for the species that we're finding today. So we revised these 29 type collections that had not been revised before. And just based on morphology, unfortunately for most of them, for all of them, even though we attempted molecular data, it was not possible. But just based on the morphology, on the morphological characteristics, we can place 10 of the species in section Hispidoderma, and we can place them down to the species complex level, just looking at the morphological characteristics. We know that those names belong in a certain species complex. 10 in section Hispidoderma, 12 in section Celuloderma, 2 species in section Pluteus, and there were 5 species originally described as Pluteus that were not Pluteus at all. So now, at least when we deal with Pluteus, we can forget about those names. In North America, we have about 100 species-level clades in the phylogenies for Pluteus. For 46 of them, we can put a name on them. For 54, we cannot. And this number has gone up. I did this presentation last year, but I didn't want to change the 100 because it's such a nice round number. We're probably now up to 56, 57 undescribed species. There are more undescribed taxa in North America than species we can put a name on. This is one of the examples where we can resurrect an older name, which is such a satisfying process, being able to say, yes, this is what Muriel described 100 years ago. Now we have modern collections. Now we have DNA data for it. This is Pluteus abelanius, which is a species that Muriel described from the Adirondacks in New York. This is a collection actually from Quebec. And this species is quite widespread in Eastern North America. And it's a case where just looking at the type collection, we're able to make that connection just using the morphological characters. I'm going to tell you about a couple of the papers in section Celuloderma that were just very recently published. This was published in 2022. And we were trying to do the same thing. Look at older names, see which names can be used, how to connect those names to phylogenies. And this was the Pluteus romelii clade. And the name Pluteus romelii was basically used for any small Pluteus with a brown cap and a yellow stem. If it had those characters and it was in section Celuloderma, that was Pluteus romelii. And nobody looked much further than that. There was a species from Muriel described from the West Coast, Pluteus fulvipadius, that some people consider a synonym of romelii, some people were not so sure. And looking at the phylogenies, sure enough, they were right to doubt that was actually the same thing. Pluteus romelii in the strict sense, and we just selected a neotype collection. So now we have a reference sequence for where Pluteus romelii is. And in this is this clade that we highlighted here in yellow in the phylogeny. In North America, we have 3 other close relatives of Pluteus romelii that are different. And one of those relatives is that name from Muriel, Pluteus fulvipadius. We have Pluteus fulvipadius here, Austrofulvus that we describe as new, Parvisporus that we describe as new, and Bellingi that we describe also as new. And Pluteus fulvipadius is something that Muriel described from Oregon more than 100 years ago. It's present in all over the West Coast, from California to BC. It's also present in Eastern North America, but doesn't go much further south than the Great Lakes. It's recorded from Michigan, Quebec, Minnesota. It's relatively big for a Pluteus. And one morphological characteristic is that the gills also stay yellow until eventually they turn pink because the spores being produced in the gills change the color. In Western North America, sometimes the very young specimens can have these green colors on the gap. It's really beautiful, really striking, but we actually have sequenced specimens like this, and it's just a morphological variant. We then described 2 new species for which there were no names available. This is Pluteus austrofulvus, which occurs only in Eastern North America, but farther south than Pluteus fulvipadius. The gills are also a different color. They're always white, and they stay white until eventually they turn pink. And this is known mostly from Arkansas, Georgia, Indiana, so a little bit south than fulvivarius, and that's why we name it Austrofulvus. Austro means southern, so that makes reference to the relative distribution. And this is Pluteus parvisporus, which again, you look at the morphology, they all look so similar to each other. Brown caps, yellow stems, is when you have the guidance of the phylogeny that you can go back and start detecting those small differences in spore size, coloration of the gills, geographical distribution. Once you have the guidance of the phylogeny, it's easier to go back and figuring out where the species belong. This is Pluteus bellingi that was dedicated to Elsa Bellinga. Elsa is a mycologist originally from the Netherlands, but she has been in California for over 2 decades now. And she actually collected the type collection. And this is look at the morphology again, so similar to the photos of the species that I just showed you. Those species are here in the phylogeny. Pluteus bellingi is down here. So molecularly, you can look at things that are so different, but the morphology, you really have to go to the teeny tiny details, spore size, cystidia shape, and even then sometimes you end up with very few morphological distinctions between species that in the DNA analysis are clearly different lineages. The other paper that was recently published, and it was just last year, it was again section celluloderma. In this case, this group of species, they have rounded cells in the cap, but they also have intermixed these elongated cells. And this is what in traditional taxonomy was called Pluteus potospilius, which is a European species originally described from England. Again, no type collection, so no way to generate a reference sequence. And in North America, there were only 2 species that were considered to exist in this group, Pluteus sediceps and Pluteus sediceps parietis stidiosus. Well, the phylogeny is still a very different species, a very different story in terms of species diversity. This is the clade that we decide corresponds best to Pluteus sediceps, and this is the type collection of Pluteus sediceps parietis stidiosus. You can see in the phylogeny, they end up in completely different places. They're not related to each other. And Pluteus stidiosus itself is a good species in its own right. So we raise the variety to species level, and it's actually closely related to Pluteus potospilius itself. So 2 names were available for North American taxa. We have 10 species, at least, of this group in North America. And in the paper, we were able to formally name 6 of them. 2 of them had names available, Pluteus sediceps, an Atkinson species that was originally described from North America, Pluteus inflatus, which is a European species that is present, and 4 taxa that were either recombined or described as new species. And this is a very funny case. It is called Pluteus inespectatus, and it's a species from René Leboeuf, and she's an amateur mycologist from Quebec. And the name inespectatus comes from the fact that we had 1 collection of this species. We were not going to describe it because we only had 1 collection, so we didn't feel we had enough data yet, even though molecularly it was different. And just before we were almost ready to send the paper for publication, René found a 2nd collection and said, okay, I'm going to sequence this one. So we sequenced it, and it's now the 2nd collection of Pluteus inespectatus, means the unexpected one. And it was described as a new species, again, based only on collections made by citizen scientists. And there are 4 species in the phylogeny they're present in the phylogeny in the paper, but we couldn't describe them because they're either known from a single collection. This is 1 collection from Ohio, 1 collection from Mississippi, 1 collection from Indiana, or the only data that we have is environmental sequences. We know that species is somewhere out there in Mexico. We have 3 environmental sequences, but we have no collections to describe the species. So we have even, again, all these projects, they have been going on for 10, 15 years. And when you sit down, write the papers, put everything together, you are still left with things that you cannot describe because you don't have enough collections yet. You need to keep collecting. You need to keep sequencing. You need to keep studying those collections before the descriptions can be made. A couple of notes to finish with the decade of molecular studies. I haven't been doing much work in Bulbo pluteus, but other people have, and many of these people are collaborators from our initial phylogenies. Bulbo pluteus michiganensis, the second record of Bulbo pluteus michiganensis actually came from Russia. And these are the photos from the Russian paper from Siberia that, that show Bulbo pluteus michiganensis growing there. And this was published in 2019. So it took almost 10 years to find another collection of Bulbo pluteus michiganensis. Bulbo pluteus asiaticus, it took even longer. Only last year, there was a second worldwide record of Bulbo pluteus asiaticus. And there was an interesting species from Mexico, Bulbo pluteus canalipes. This was not a new species, but a new combination of another very old name of Bulbariella that was being transferred to Bulbo pluteus. For Bulbariella itself, there has been no overall revision in the last 10 years. What some people have been able to do, Alfredo Vicini from Italy, which was a collaborator in the first phylogenies, just a few weeks ago, he published a paper where we finally resolved where Bulbariella belongs in the order Agaricales, which up until to this point, it wasn't clear at all. So now we know that Bulbariella is somewhat close to Pluteus, is in the same suborder, but still is not part of the same family. This is the suborder Pluteaceae in the Agaricales. This is the Pluteaceae, which is defined the same way we define it in the paper, just two genera, Pluteus and Bulbo pluteus, and Bulbariella is here. And because it's here and it doesn't fit anywhere else, it got its own family. The Bulbariaceae is going to be the family for Bulbariella moving forward. And just to finish the talk today, I'm just going to give you a very small glimpse of some of the things that we are currently doing, that we are doing right now, and that are working on. For the papers that we just published on the species diversity of Pluteus, I told you about these guys here and these guys here. Each one of these color boxes is its own project, is its own paper. And some of them, and you can see by the size of the boxes, are going to be much, much bigger projects. One of the projects has about 70 species to be described, both older names and new species. So we have some big projects to finish between this year and next year. I'm just going to tell you about one of those projects because it's almost ready to go for publication. This is Pluteus leoninus, the lion Pluteus, and the leoninus is because of the yellow, bright yellow colors, which is not very common in Pluteus. And again, we have quite a few species in Europe that are considered either closely related, maybe synonyms of Pluteus leoninus. And in North America, we have one name, Pluteus flavofuliginus. It's an Atkinson name that has also been considered maybe a synonym, maybe different from leoninus. And this is section Hispidoderma, so long upright cells in the cap. And the cystidia have these tiny, like finger-like projections at the apex. There is not a lot of morphological variability in the Pluteus leoninus group. The colors go from bright yellow to brown, and the colors on the stem go from yellow to pinkish. And once again, surprise, surprise, the species that we thought we might have in North America, we don't. Pluteus leoninus itself, it's only present in Europe and Asia. Pluteus flavofuliginus, which was originally described from New York, is actually the correct name for the Eastern North American version of Pluteus leoninus. And again, we were able to sequence the type collection, and it's a type collection from 1908, I think. So more than 100 years old, we were able to get a partial sequence for that. And we end up with the Western North American lineage that you can see here. This is kind of funny because morphologically doesn't look like the other lineages of Leoninus, looks like a different species in this group. And following the theme of big cats, since this is Pluteus leoninus, the Western North American version is going to get the name Pluteus pumae for the puma. We also have a couple of new species in Eastern North America in this group. This is Pluteus aureus, another bright yellow species, and this is Pluteus croxeus. Croxeus makes reference to that orange-red color that you see on the cap there. And before we described those species as new, we went back and said, well, is there any older name that we can actually use for these lineages that we see in the phylogeny? So with the data that we had, we ended up concluding that 4 of the species that Muriel described, and when I say describe, the descriptions of Muriel look something like this. That's all you have other than the collection itself. 4 or 5 lines of morphological description where the type collection is, and then whatever characters you can gather from the type collection. Muriel described 4 species from Eastern North America, but we ended up concluding that all those 4 species are actually synonyms of Pluteus flavofuliginus, which is the older name in this group. For Bulbopluteus, again, iNaturalist, citizen scientist, that's the future of biodiversity science. I think this is the 3rd collection, the 3rd North American collection of Bulbopluteus michiganensis, the 2nd after the type. It was collected in 2023, and now we have a sequence for it, and it came through citizen scientist sequencing projects. And now we have an actual photograph of what Bulbopluteus michiganensis looks like, a North American collection for it. Bulbariella is gonna take a minute before we get to the North American species. We have been working on a revision of the European species for the last 10 years. We now have almost 350 barcode sequences, and the diversity is just astonishing. It's just, we have only 13 of those lineages, of those clades that we can name. 20 species are going to be described as new just from Europe alone. We haven't even started with North American species. So it's going to be a minute, but hopefully this Bulbariella big revision is published this year, and then we can move on to the North American species. And that's all I wanted to tell you today about the Pluteaceae and all the work that we have been doing and we are doing. And I will be more than happy to take any questions.
Thank you so much. That was very interesting. It was also great to hear about like all the ways that citizen science is contributing to this. So, all right, it looks like we have some cameras on. I don't know if that's for questions. Kyle, I saw you first. Did you have a question? No, just showing your face. Okay. Yeah, so again, if you want to use raise hand or just go ahead and unmute if you have a question. Or if you'd like to type it in the chat, you can type there and we'll read it out.
Just wanted to say I saw you in Canada at the NAMA, and it was great. And I'm glad to see new stuff is happening all the time. Glad to see what you're doing.
Thank you.
Maybe while folks are gathering their thoughts, I had a question. So you mentioned the specimen from like 1932 that you were able to sequence, and it sounded like maybe it was barcodes.
Yeah.
I'm curious, like how, if you have been working on sequencing older fungus from collections and like maybe trying to do more reduced representation, genomic things, and like how that's going.
I haven't been doing it myself, but
Yeah,
I know it has been done. And people have been doing it with even quite old herbarium collections. And sometimes people want to get, I know people at Kew in London have been doing that for the Fungarium. And sometimes they just want to get broad representation of genes from herbarium specimens of any age. And sometimes they're targeting older specimens and just see what they can get,
Yeah.
because they know traditional methods are not going to work. So you just try next generation sequencing and see what genetic information you are able to gather from all the specimens. And it so far is working relatively well. There are some groups of specimens that are just too far gone for anything at all, but I think people who are doing next gen sequencing directly on herbarium specimens, they get pretty good results. So maybe in the near future, we try again for some of the mural things that didn't work. We may go back to them and try next generation sequencing, and we see if we got it right the first time.
Yeah, go ahead, Kai.
I was just going to say, I really enjoyed learning about the Pluteaceae family tonight. It was I've been reading up on mycology in general just for the past year or so. It's become like a hyperfixation for me, but I have no real like grounding in like actual families. So this is a great little like starting point to actually get down to like the nitty-gritty of like this whole family and this whole wide range of taxonomy. I was fascinated by the variations of just like what was it, a single species?
Yeah.
Mhm. It's great. It was great to get like a good starting point into like furthering taxonomy knowledge. I was actually curious because it's I believe you mentioned at the beginning it's a saprobic type of
Mhm.
mushroom, the Pluteaceae family. I was wondering because I've been that when I first started getting into mycology, I was drawn into the practice of mycoremediation, cleaning up toxins in the
Mhm.
soil, heavy metals and whatnot. I was wondering if this is an ideal candidate, because it's
Probably not. And there's something really funny, not funny, haha, but funny, frustrating for scientists about the Pluteaceae that you cannot grow them in culture, which is very weird for saprotrophic mushrooms because they're pretty happy to grow if you give them a substrate that they're happy growing in. But Pluteus does not grow in culture. Bulbariella, which is not now in its own family, it can be cultured and some of the species are edible. So that could be more of a candidate for something like mycoremediation. And I think anything that we grow, that we have learned how to grow in the lab really well, like Agaricus, the button mushroom that we know, Bulbariella bulbaceae is actually grown for food especially in Asia. So things that we know pretty well how they grow under lab conditions, I think they would be the better candidates for anything like mycoremediation because you have a good sense of their biology in terms of what they need to grow. And you want to optimize the growth to then release them into those environments that need the mycoremediation. So maybe Wolvariella could be a candidate in the future. Plutus, unfortunately, it just doesn't want to grow when we try to grow them in the lab. They want to keep their secrets. They're
very
not
stubborn.
they're stubborn. They're like, no, you have to figure out the taxonomy yourself. I'm not gonna grow.
All right. Well, thank you. That's a great I'll look into that one a little bit more. Thank you.
All right, I'll give just another 30 seconds. If for any questions, again, you could just unmute or put your question in the chat. I have a final question that I like to end on, so we will have at least one more, but let's see if anybody picks up in the meantime. Okay, Well then, to take us out, I like to end all of our talks with asking the speaker, like, if there is one takeaway, like one lesson to learn from what you've been talking about today for people to walk away with, how would you like to distill it down? What would it be?
Citizen science is the future. Study the things that grow in your backyard, and you have undescribed species in your backyard. This is my takeaway message: is diversity, fungal diversity is everywhere. When we talk about diversity and describe species, sometimes we tend to think of exotic places in the tropics, which are very important and need to be studied, and they have a lot of diversity. But fungi, they're really literally undescribed species of mushrooms in everyone's backyard.
Oh, all right. Well, let's all head outside and look for the mushrooms. All right. Thank you everyone for being here. I hope you all have a good night. Keep an eye for announcements for our future events, and make sure to check out torreybotanical.org. Have a good night, everyone. Thank you.
Bye.